In this section, I'm going to talk about bacterial growth incubation. I talked about how bacteria require particular media in order to successfully grow, and some bacteria, no matter what you give them, are just impossible to culture in a lab. So we depend on investigating them using genetic identification tools. Incubation is another thing that you have to take into consideration when growing microorganisms. Incubation requirements are specific depending on the species. Although many microorganisms that we care about as people tend to grow at 37 degrees Celsius, which is equivalent to 98.6 degrees Fahrenheit, our body temperature, and many of them do grow in the presence of oxygen, there are some specific requirements that allow microorganisms to grow better. So we need to consider this when we're trying to culture microorganisms in a laboratory. So incubation is talking about the growth of microorganisms in a particular environment. So we inoculate the media and then we have to incubate it or allow it to grow, and we put it in a certain environment in order to do so. In a laboratory, this would be an incubator. An incubator can also control atmospheric gas in addition to just temperature. It controls the amount of oxygen and carbon dioxide the microorganism is exposed to. Microbial growth in liquid medium, you can see cloudiness, sediment, scum, color. Microbial growth on solid medium, you're going to see visible masses of piled up cells, which are colonies, or you can see lawns. Incubation conditions of various bacterial pathogens. So Listeria monocytogenes grows from 25 to 30 degrees Celsius. Growth at 37 degrees will be pretty weak. Culturing time is 1 to 2 days. Pseudomonas is similar to Listeria. Streptococcus pyogenes and Mycobacterium tuberculosis require hotter temperatures. But you can see the culturing time for tuberculosis is 28 days as opposed to 1 to 2 days, which is pretty typical. Mycobacterium leprae, M. leprae cannot be cultured in vitro. In its animal model, it exhibits one of the slowest reproduction times, 14 days, of any known bacterium. So we can't just culture it in a laboratory. We have to have it in an animal model. There's temperature ranges that we have to take into consideration when trying to grow microorganisms. Minimum temperature, maximum temperature, and optimum temperature have to do with the temperature range. So minimum temperature is the lowest temperature that will allow that microbe to grow. Maximum temperature is the highest temperature that will allow that microorganism to grow. If an organism goes below its minimum temperature, its metabolism will essentially stop. This doesn't mean that it will die, and oftentimes we can preserve microorganisms by decreasing their temperature. Maximum temperature, though, will denature proteins, so it'll unfold proteins and permanently inactivate those proteins, and this will cause cell death. The optimum temperature is the best temperature that this microorganism will grow at. So most clinically relevant pathogens, their optimum temperature is 37 degrees Celsius, or body temperature. This promotes the fastest rate of growth and metabolism. And here you can see various categories based on the temperatures that they grow in. So a psychrophile grows in the coldest temperatures. Psychrotolerant grows at room temperature or a little bit colder. Mesophile contains those human pathogens. Those are going to grow best at body temperature. Thermophiles grow in warmer temperatures, and extreme thermophiles grow in extremely hot temperatures. You should be familiar with the organization here, not necessarily the exact temperature. So know that psychrophiles are the coldest, psychrotolerant are going to be the next coldest category, and then mesophiles, that's where a majority of the human pathogens are going to be. Those grow in moderate temperatures. The thermophiles grow in the next hottest, and extreme thermophiles are going to grow in the hottest temperatures. In addition to controlling the temperature that we're going to incubate them at, we need to consider their oxygen requirements. There's aerobes and anaerobes, and then there's a variety in between. So aerobes, aerobic organisms, require oxygen. so they can use gaseous oxygen in its metabolism. An obligate aerobe cannot grow without oxygen. People are obligate aerobes. Now we can survive for a short period of time without oxygen, but then we're going to need some oxygen. We need to produce way more energy than we can sustain on just the energy produced by glycolysis. So as people, we're obligate aerobes, but there's many microorganisms that are obligate aerobes as well. A facultative anaerobe does not require oxygen for its metabolism, but it's capable of growing in the absence of oxygen and presence of oxygen. It metabolizes by aerobic respiration when it has oxygen, and then it goes through fermentation when oxygen is absent. So a facultative anaerobe prefers oxygen. It can use oxygen to produce energy, but it can also grow without oxygen. Now, people can't do this. We can't survive without oxygen for a long period of time. Bacteria, there's one cell. They don't really do a lot. Many of them aren't even motile, so they just kind of hang out and do their metabolic processes. They're just fine being able to adapt. So many human pathogens are going to be facultative anaerobes. A microaerophile does not grow at normal atmospheric concentrations of oxygen. They require a small amount of oxygen in its metabolism, and they usually live in a habitat that provides them a small amount of oxygen, but they're not directly exposed to the atmosphere. So microaerophiles can live in like swamps just underneath the oxygenic zone of the water. So they're going to be near oxygen, just not in direct contact with it all the time. An anaerobe, an anaerobic microorganism, lacks the metabolic enzyme system for using oxygen in respiration. Strict or obligate anaerobes cannot tolerate free oxygen and will die in its presence. Oxygen can be toxic, and there's oxygen compounds that are oxidizers that are really harmful to cells. So some cells don't have the capability of handling that, so they can't be near oxygen. So here are some tubes that show oxygen utilization in different tubes. So here you can see that there's absolutely no growth except for the top. This is an obligate aerobe. Here you can see that there's a lot of growth toward the top and then some growth here at the bottom. These are going to be facultative anaerobes. Here you can see that there's pretty much equal growth throughout, and those are going to be the aerotolerant anaerobes. And then here you can see the growth is nowhere near the oxygen, and these are going to be the obligate anaerobes. How do we culture anaerobes? Well, you can do it in one of these CO2 containers. So you're going to put the plates in there, or all the media in there, and then you put an oxygen indicator and an oxygen packet in there, which will change the atmospheric conditions to increase the carbon dioxide concentration, and this creates an anoxygenic environment. And then you seal it up, you can put it in the regular incubator and allow it to grow. Another way to culture is using a hood like this. So you would put your stuff into a chamber, and then you close that chamber and push a button, and that will exchange the atmospheric gases inside so that they're anoxygenic, or without oxygen. And then you can put your arms through here, and you're on the outside where there's oxygen, but the inside of here has very low concentrations of oxygen or no oxygen. So then you can pull everything out into this chamber here, and you can start doing your culturing. Aerotolerant anaerobes do not utilize oxygen but can survive and grow in its presence. They can tolerate oxygen. They're not harmed by oxygen because they possess alternative mechanisms for breaking down peroxides and superoxide, those toxic chemicals that can be produced by oxygen. Capnophiles like carbon dioxide. They grow best at higher carbon dioxide tension than is normally present in the atmosphere. It's important in the isolation of some pathogens. Incubation is carried out in a CO2 incubator that provides 3 to 10% CO2. 5% is pretty standard. Strep pyogenes is an aerotolerant that loves or grows best in a carbon dioxide incubator. So we're going to grow strep species in a CO2 incubator. So we talked about the different growth requirement or incubation requirements. So we had temperature ranges, we had oxygen requirements. Now we have to consider pH. Some microorganisms love to grow in the presence of acid. Some of them love to grow in the presence of a base, and others need to be in a neutral pH environment. An obligate acidophile requires an acidic environment for growth. An alkalophile or alkalinophile requires a basic environment or high base in order to grow. We can also consider osmotic pressure. So osmophiles live in habitats with high solute concentration. Obligate halophiles require high concentrations of salt for growth. Facultative halophiles are resistant to salt even though they do not normally reside in high salt environments. And then we can consider hydrostatic pressure. Barophiles are deep sea microbes that exist in pressures up to 1,000 times atmospheric pressure. They're so strictly adapted to high pressures that they rupture when exposed to normal atmospheric pressure. So if you want to study barophiles in a laboratory, you're going to have a high pressure environment to allow them to grow.